In Chapter 4, Part 2, we will continue to discuss the connection between addiction and the nervous system by taking a closer look at specific cells and chemical pathways. This is also the area where new research continues to be conducted, which helps increase our understanding regarding the complexity of addiction within the nervous system. Two types of cells are found within the nervous system. We will start first with glial cells because these have generally received less attention, as they were first believed to be unimportant in the study of addiction. That has changed as there is now more understanding regarding the importance of glial cells. These are cells which make up between 80 to 90% of the brain. Initially, they were thought to take a back seat to the flashier neurons, but we now know the vital contributions of these cells. Listed on this slide are the specific contributions. Please note, glial cells are now recognized as playing a central role in addiction and mental health, and more research continues to be conducted in this area. Moving on to the neurons, these are the cells that are responsible for analyzing and transmitting information. As presented in the text, the specific regions all contribute to the communication, known as neurotransmission, that occurs between neurons. Stored within neurons are neurotransmitters, which are viewed as vital chemical messengers. Looking specifically at addiction issues, there are several neurotransmitters that can be impacted. As an introduction to this area, we are going to look at three. These are dopamine, serotonin, and norepinephrine, and are considered to be the trifecta of neurotransmitters with addiction. These three have the capacity to overlap and interact in ways that significantly impact areas such as mood, attention, and cognitive functioning. When looking at the impact addiction can have on the neurotransmitters themselves, the general impact occurs both in the intoxication and withdrawal process. What we see here are the effects that occur on neurotransmitters during the intoxication process are generally described as the opposite of what occurs in the withdrawal process. For example, if the intoxication process causes a release of a neurotransmitter, the withdrawal process will result in the opposite effect. When discussing withdrawal and the impact on neurotransmitters, we need to keep in mind both acute withdrawal, again acute meaning in the present and shortly after ceasing use, and what is called post-acute withdrawal syndrome, or PAWS for short. During PAWS, some symptoms commonly reported are anhedonia or lack of pleasure, difficulty with concentration, as well as problems with memory, issues with mood stability, depression and/or anxiety, struggles with coordination and clumsiness, and sleep disturbances. This again is an area where more and more research is being generated to fully understand the brain's healing process. For PAWS, depending on the type of substance or substances, the length of use, as well as a variety of other factors, symptoms of PAWS, as well as the brain's healing process, can often be viewed as at least a 3 to 6 month process, not uncommon to need 9 months to 2 years to achieve optimal functioning levels. Now, let's take a closer look at each of these three neurotransmitters. Starting first with dopamine, it is considered to be the neurotransmitter that is affected by all psychoactive substances, as well as a key component in behavioral or process addictions. Dopamine is a component of the reward properties discussed earlier by stimulating the reward pathway. However, please be aware that more research and information is needed to understand the complexity involved with this system and dopamine itself. Next is serotonin, which is connected to areas such as mood, sleep, desire, and impulse. While this neurotransmitter is often impacted by many substances, in particular, it is heavily at work with stimulants and certain hallucinogens. The last of the three is norepinephrine, which again deals with mood, but is also involved with movement, memory, and sensory processing. More specifically, it is a major contributor to the fight, flight, or freeze response, as well as general anxiety responses. It can be an asset because it helps us to keep us alert and able to act in what may be perceived as a dangerous situation. Lastly, we want to understand the specific ways psychoactive substances can impact the neurotransmission process. While there are many possible ways, there are three main ways. The first is to mimic or increase neurotransmitter activity, which is described as being an agonist. Heroin, morphine, as well as nicotine are considered agonists. The second way is to block or reduce neurotransmitter activity, which is being an antagonist. An example of this would be naloxone, which is used to treat opioid overdose. The final way is what we refer to as a reuptake inhibitor. During the neurotransmission process, some residual neurotransmitters can be left in the synapse, or the space between neurons as described in the book. These neurotransmitters will try to make their way back to the original neuron to be reabsorbed. However, with a reuptake inhibitor, this reabsorption is initially blocked, leaving an excess of neurotransmitters to remain in the synapse. An example of this would be cocaine.